Welcome everybody to ICPR 2020. This year is an online version. My name is Tobias Buchborn. I'm a biopsychologist and I'm currently with Imperial College London. And today we would like to talk about why is it actually that when you take a drug repeatedly that the drug would change its face over time. If you take a drug for the first times, you might have, for example, your euphoria. And if you go on to take the drug on and on, this euphoria wears off, so you become tolerant. Other effects of the drug, on the other hand, might sensitize, so they become more and more pronounced. For example, the craving for the drug. Sometimes it happens at some time that new effects will come into the arena which had not been there in the first place. And if you decide to stop taking the drug, you sometimes end up in a withdrawal syndrome, which very often in its symptoms is the sheer opposite of what you experience in the acute state. Of course, not all drugs are the same, and LSD, for example, would not check all of these boxes here, but what is it actually we do know about the repeated application of LSD? We do know that it's actually quite uncommon to take psychedelic doses of LSD at high frequency. As to this, withdrawal syndrome is also unheard of. Something that is very prominent with LSD is tolerance. Probably a reason why people don't do it that often. If you take psychedelic doses of LSD 4 days in a row, you are next to completely tolerant by the fourth day. And as quickly as it comes, it also seems to go. 3 to 6 days off of LSD seems to be sufficient to reignite the original intensity of LSD experience. Back in the days, they applied LSD even for 84 days. And by the end of this time, they exchanged LSD to a placebo, and the subject would not even recognize that it was exchanged. That rigorous tolerance seems to be. You should keep in mind, though, that these human studies were done back in the days, and they don't really measure up with modern standards. So there's still a lot we don't understand, we don't know about tolerance to LSD, and that's why we wanted to add a little bit to the understanding. Particularly, we were interested in the mechanism of tolerance. A very important mechanism is the pharmacodynamic one. And it refers to the fact that the target receptor of a drug, after some time, would stop responding to the drug. This is called desensitization, and there are different phases, factors that contribute to such a desensitization. The receptor might be uncoupled from its signal cascade, it might be removed from the membrane, internalized, and it might be degraded, and the synthesis of the receptor might be inhibited. We know that LSD psychedelics act primarily by activation of serotonin 2A receptor, and we also know from animal studies that this 2A receptor can form complexes with the mGlu2 receptors. So if we're interested in tolerance, then it makes sense that we look at these receptors and adaptation of these. This is what we actually did. You see the overall project. We will not be able to speak about all of this today, just to give you an idea how complex this phenomenon indeed is. When investigating tolerance in animals, of course, you cannot really ask the animals for their conscious experience. That's why you need to look at behavior output and somatic markers. We here decided for 2 animal models, the shaking behavior. Let me show you how it looks. Yes, how it looks, and for hypothermia, increase in body temperature. We chose these models because they are primarily related to the serotonin 2A receptor, as is psychedelia, and we can find both in humans as well. We started off with the dose response curve. We wanted to use the smallest dose possible, but of course still see something. And we learned that when we use this small dose and we put the animals together with their familiar littermates, they would respond better. That's why we continued that way. We used this dose, reapplied it at 4-hour, 8-hour, and 24 hours interval, and learned that the longer the interval is of the reapplication, the less likely animals become tolerant. We then went on to apply LSD on several days in a row. If you apply LSD once or twice per day, animals would not become tolerant. Only if you increase the dose at 3 occasions during this regimen to medium level, they would become tolerant. So there's a dose dependency here. We also have similar results for DOB, which is another serotonergic hallucinogen. We're then interested in whether different effects of LSD would show different tolerance development. We learned that LSD would increase the temperature, body temperature of rats when the environmental temperature is higher than the usual room temperature. And this effect can be blocked by ketanserin, so it's a serotonin 2A specific phenomenon. We then applied LSD in the same way. We found tolerance for the psychedelic signature move, for the shaking behavior, but we learned that animals would not become tolerant to hypothermia. Only if we increased 2 additional dosings during this regimen, they would become tolerant. So there is again a dose dependency here and obviously a biased tolerance development. We then had to look into the frontal cortex because it's very important for the psychedelic effect, but also believed to be important for the psychedelic signature move. We had to look at the serotonin 2A receptor and at the mGlu2 receptors, because we know that the primary output of serotonin 2A receptors in the frontal cortex is glutamate release. We also had to look at the overall glutamate receptor binding. In DOB-tolerant animals, we found that, as is expected, that both the serotonin 2A binding capacity but also its capacity to bind G proteins is decreased. We also found the desensitization of mGlu2 receptors, which is interesting because DOB would not directly bind to it. For LSD, unexpectedly, we didn't find any significant changes in serotonin 2 parameters, serotonin 2A parameters, but we also found this mGlu2 desensitization. Interestingly, as opposed to DOB, we found a decrease in overall glutamate receptor binding. So it appears that there are different ways of how the brain achieves tolerance, either directly by adaptation of the 2A receptors or by adaptations of downstream events. Interestingly, in the context of the hypothermia experiments where we applied higher doses of LSD, we did find serotonin 2 desensitization. So again, we have a dose dependency here in terms of tolerance-related phenomenon. Tolerance aside, we were interested in whether there are some therapeutic implications of the repeated application of LSD. We used bulb-actomized rats here. These animals have a disrupted limbic system, and the limbic system is very important for allocating emotional valencies to the environment and memorize the environment accordingly. And if this system is disrupted, animals, for example, are not able to avoid an aversive situation, although there is a way of avoiding it. How is this measured? You see it in this overall setup there. An animal hears a tone, and after this tone, a current is being applied. This current is not painful, it's not harmful, but it's a strong tickle, and animals would rather want to avoid it. And whenever the animal hears the tone, it actually can escape the currents by jumping to a pole. The bulb-actomized rats, however, are not able to do that. But if we give them antidepressants subchronically, they become able to learn and to escape the aversiveness. Therefore, we were interested in whether LSD would do the same. And indeed, it did. Interestingly, the healthy animals that received LSD did not benefit from LSD. So this seems to be a specific effect, an ameliorating effect for the pathological condition. We also had a look into the brain, this time into the hippocampus, because it's part of the limbic system and it's very important for learning. We found that bulbectomy is associated with the desensitization of serotonin 2 signaling here, and that LSD would normalize this. Again, in the healthy animals, LSD rather desensitized the receptors, so the effect of LSD is again specific for the pathological condition for the depressed animals here, this normalizing effect. Serotonin 1A signaling and overall metabotropic serotonin signaling didn't show any regulatory activity here, so it was something specific to the serotonin 2 family. our research aside, we'd like to discuss with you a basic phenomenon or parameter of pharmacology. It's called affinity, and actually it refers to the concentration of a drug in the synaptic cleft that is needed in order for the drug to bind to a receptor and induce activity. But let me explain how we measure it. If you're interested in the affinity of psychedelic X for the serotonin 2A receptor, you would have these little wells, we call them. There are a lot of cells in there, and we would apply a radioligand to it. And this radioligand would bind all the serotonin 2A receptors. We then apply a little bit of psychedelic X, and this displaces the radioligand from the receptor. We would then increase the dose, the concentration of psychedelic X, until no radioactivity is left. And then we can draw these nice graphs here, and by convention, the affinity of psychedelic X is the concentration in the synaptic cleft, or in this well here, at which half of the radioligand is being displaced. This is the affinity. Why am I telling that? Because it's important for microdosing. It's been suggested that you need about 40% of your serotonin 2A receptors occupied for a microdose effect to occur. The thing is now, psychedelics are not selective for the serotonin 2A receptor. Our psychedelic X, for example, prefers the serotonin 1B receptor before the serotonin 2A. And this means if you microdose yourself and you want 40% of your serotonin 2A receptors occupied, then you will have about 80% of this other receptor occupied. And then you would have to ask yourself, what is it actually this other receptor does? Where is it? In the brain, in other organs of the body? And these are important considerations for safety implications, especially for long-term application. Taking home message. What have we learned today? Tolerance to LSD depends on the interval of application. The smaller the interval, the more likely animals become tolerant. We also learned that tolerance depends on the dose. The higher the dose, the more likely tolerance occurs. And we also learned that tolerance develops differentially. That means some of the effects would become more easy tolerant than others. And this again is important for safety implications and side effects and how they would develop over long term. We also found in terms of mechanism that tolerance is associated with desensitization processes which affect the serotonin 2A, serotonin 2 family, but can also be complemented by adaptations and downstream events and interaction partners such as within the glutamate system. Short-term application of LSD might have beneficial effects when wanting to integrate something, when wanting to integrate aversiveness, when going through things you have experienced and want to learn how to behave next time in a better way. And as to this, of course, this might be relevant for therapeutic approaches with these drugs and depression, but also in other disorders of the psyche. The last slide actually taught us scientists that when we do microdose studies, it's probably a good idea, especially if they are long-term, months perhaps, that we don't just measure psychological output, but also look at other organs which express receptors the given hallucinogen psychedelic has high affinity for, to see whether there are adaptations and whether side effects might be expected as to these physiological processes. That's it from my side. Some thank yous, particularly to the Institute of Pharmacology who made all of this happen, and to my doctor father, Volker Höld. Thanks for all your support, appreciate it. If you are interested in tolerance to LSD, please read our review, it's very comprehensive. And if you're interested in my research in general, please follow me on ResearchGate, and you will be updated as to all research that is coming out soon. Thanks for your attention, and take care.